In our development unit, we've seen how gene regulation explains how cells respond to the environment. We've also seen how gene regulation explains why organisms with very similar genes may have very different traits. In this video, we will take a look at how gene regulation explains embryonic development. In other words, how a zygote becomes an organism with specialized cells, tissues, and organs in all the right places. Let's take a quick look at how you developed when you were an embryo. Day 1, you were a zygote, a fertilized egg. By day 6, you were a ball of cells, a blastula, but these cells hadn't specialized yet. You were still just a little blob. By day 10, you were a gastrula, an early embryo, and some of your cells were specializing. By day 35, you were a full-fledged embryo. Even more specialization, you can start to see a head and limbs at this point. By day 63, an early fetus, even more specialization. By day 140, a late fetus, and voila, day 280, a fully fledged infant. So how did all this happen? How did you go from a cell to a baby with all those parts? Well, here are some processes involved. The main process is differentiation. This is when different cells activate different genes and thus produce different proteins. And those are specific to the type of cell they are. For example, here we have 2 different cells. They came from the same cell though. All of these cells have the same DNA. But this cell is expressing a gene that helps it become a neuron, and this cell is expressing, turning on, a gene that helps it become an epithelial cell. And in order to turn on those genes, as we know, signal transduction pathways are necessary to activate the transcription factors that will cause transcription and translation of the genes. Now, in embryonic development, those signals involved in signal transduction pathways have a special name. They're called morphogens. And they look similar to other signaling molecules, but there's a few unique things about them. First of all, they tell specifically a cell what to become, what kind of tissue, what kind of organ. And these signals, these morphogens, can be found in the unfertilized egg cell, and they're also found later in embryonic cells. So as you can see, the fate of cells, what type of cell they become, is determined very, very early in your life, and there's usually no going back. The other great thing about morphogens is that they're influenced by concentration. The concentration of morphogens in the environment will help influence their effect on the cells. For example, this group of cells is exposed to a very low concentration of morphogens, and because of that, they specialize and become epithelial cells. These 2 cells are exposed to a slightly higher concentration of morphogens, and so they are triggered to become skin cells. And this group of cells are exposed to a high concentration of morphogens, and that signals them to become neuronal cells. Let's take a look at how morphogens work in that initial egg cell. Here's the egg cell. And as you can see, the morphogens are found on one side of the egg cell. This is known as a concept of polarity, an uneven distribution of morphogens. And these morphogens, these molecules, might be mRNAs or they might be proteins, but ultimately they act as signals. Some common examples, some famous examples, include bicoid and caudal, and we'll find out more about those in class. So what these signals do is they tell early embryonic cells whether to develop into the anterior, the top, the head of an organism, or the posterior, the bottom of the organism. And so here we have what is known as the anterior-posterior axis. And again, it's the concentration of these morphogens that determine whether this part of the cell is going to eventually become the head and whether this part will develop into cells that become the bottom. And here's how that works. There's the initial egg cell with all the morphogens over here. So when this cell divides to become 2 cells, this cell has all the morphogens. Cell division continues with this cell containing the morphogens, and ultimately this part of the embryo is going to become the head, the anterior, and this part of the embryo without those morphogens becomes the posterior or bottom. Now, there are other morphogens involved. Here we have an embryonic cell known as an inducing cell. The inducing cell sends signals to a responding cell, and the signals that the inducing cell sends tell the responding cell to differentiate, to become a specialized cell. And they do that by stimulating a signal transduction pathway in the responding cell. And that signal transduction pathway culminates in transcription factors that then turn on or off genes, causing differentiation. Now, there are two main ways that embryonic induction can happen. Here's one way. Two cells can physically contact each other. Here, the inducing cell has its morphogen, its signal, on its cell membrane, and it's going to bind to a receptor protein on the responding cell, and then the responding cell will differentiate. Or the inducing cell might release the morphogens, the signals, into the extracellular fluid, and they may diffuse across to a nearby responding cell and bind with the receptor protein and start differentiation. Now, this process is involved in something known as morphogenesis. If you were to break down morphogenesis, morpho means shape and genesis is to create. So morphogenesis is when the cells specialize at a point where we're starting to see the shapes of organs and tissues. So morphogenesis is developing tissues and organs with the correct shape, a round head, a hand with 5 digits, and so on. And this involves not just cell division, because we need more cells, we also need differentiation and migration and even death of cells, and we'll find out about that in a second. A similar process that occurs is pattern formation. So this is just what it sounds like. This is the development of tissues and organs in the correct position or place. So a head at the top of the body, limbs along the middle of the body, and depending on what kind of organism you are, a tail in your posterior. Now, what controls these patterns? What makes sure that your arms develop in the right spot and don't grow out of your head? Well, there are something called homeotic genes, or master genes. Here, if we look at the chromosome of a fly, these are all homeotic genes, and they are going to control pattern formation. And it's pretty simple. The order that they occur on the chromosome is the order of the pattern that will occur in the embryo. One very famous example of homeotic genes are Hox genes. We'll find out more about those. And so what these genes do is they determine what each part of the embryo becomes along the anterior-posterior axis. So this homeotic gene determines that this part of the embryo will become this mouthpiece on the head. And then this green homeotic gene will determine that this part of the embryo becomes the head. Similarly, the brown homeotic gene determines that this part of the embryo becomes the mid-region or the thorax of the fly. And the homeotic genes act by coding for transcription factors that tell the cells, again, how to differentiate. And the cool thing about homeotic genes is that they're found in many, many organisms, and they're highly conserved. They're very similar. So this fly's homeotic genes look very similar to a mouse's homeotic genes, and they look pretty similar to a human's homeotic genes. Within a homeotic gene, there's a special region known as a homeobox. This is a sequence of 180 nucleotides, and it's found in all homeotic genes. And what it does is that those 180 nucleotides code for 60 amino acids that are found in a transcription factor. And those 60 amino acids are crucial because they are the binding site for the enhancer region. Remember that transcription factors bind enhancers to promote transcription. Now, if we disrupt homeotic genes, if we disrupt these master genes, crazy things can happen. For example, this is the result of a transplantation experiment, which scientists do to study development. What happened here is by interrupting the homeotic genes, a fly that should have antennae growing out of its head now has legs growing out of its head, which is awkward to say the least. So you can imagine that if mutations occur spontaneously in a developing organism and they disrupt homeotic genes, it can have serious effects in development, in fact, often fatal effects. The last process we're going to look at today explains why you do not have webbed hands and feet. This process is known as apoptosis. Apoptosis is a fancy word for programmed cell death or cell suicide. And this is critical to morphogenesis and pattern formation because sometimes you want cells in a certain area and sometimes, like between your fingers, you don't want cells in a certain area. And so those cells kill themselves. And here's how. Here we've got a cell, and it is going to receive signals, either from inside or outside, that are going to trigger suicide genes in its nucleus. Those suicide genes code for proteins that will kill the cell. And once those proteins are produced, the cell is going to shrink. It's also going to release some signals that will tell a nearby cell to become phagocytotic. In other words, it's going to tell a nearby cell, eat me, I'm dying. And so here we can see that nearby cell has engulfed and ingested that dying cell. And this is important because this allows the materials that were in that dying cell to be reused by cells that get to live. And keep in mind, not all cells do apoptosis, only certain cells undergo apoptosis. So this is tightly regulated, often by those microRNAs. And that concludes our exploration of processes related to gene regulation that explain embryonic development.